Hello everyone, my name is Hussain and I will be teaching you Neet Biology Chapter 2 Part 3 today from the chapter Biological Classification, which is basically your second chapter from the 11th standard NCERT textbook. Okay, in today's chapter or in today's topic we will see Kingdom Monera Part 2. Under this topic we will study the structure and motility of the bacterial cell, the nutritional diversity of the bacteria and reproduction in bacteria. Starting with the structure of a bacterial cell, the cell wall. The cell wall is an important part of the bacterial cell, as we saw in the previous class that the cell wall made up of peptidoglycan is a distinct feature of bacteria. Okay, so here as compared to plants, the cell wall is made up of a non-cellulosic material, and that is why it is termed as non-cellulosic because it's not made up of cellulose. Okay, so it is made up of peptidoglycan, which is made up of two components: a polysaccharide, a carbohydrate moiety or a carbohydrate part, and a protein part, okay, which is made up of amino acids. So yes, the peptide and the glycan part. The glycan part, the carbohydrate part, is again made up of alternating amino sugars, namely N-acetyl glucosamine shown in orange in the diagram and N-acetyl muramic acid shown in green in the diagram. Okay, now the N-acetyl glucosamine and N-acetyl muramic acid, both of them are linked together with a bond. Both of them are carbohydrates, so they are linked with the help of beta 1,4 linkages or beta 1,4 glycosidic linkages, okay, which helps to join two carbohydrate moieties. And you see in the diagram that there are three layers of peptidoglycan chains. Okay, so each chain is joined to one another only in the green molecule, which is N-acetyl muramic acid, and these are joined with the help of bonds known as peptide bonds. Okay, so within the chain between NAG and NAM, it is 1,4 beta 1,4 linkage, and between the chains, between two chains, then it is peptide bonds. Okay, next is motility in bacteria. Bacteria can be motile or immotile based on the presence or absence of flagella or based on their ability to move on. Okay, so motility is generally through the help of flagella. So based on this, they can be divided into two. A, trichus. Trichus means trichome. Trichus is derived from trichome, which means hair-like structure. Flagella is a hair-like structure. Yes. A means absence. Absence of trichome gives the name atrichus, which means no flagella, bacteria without flagella, and trichus bacteria means bacteria with flagella. So based on the presence or absence of flagella, based on the position of the flagella, and based on the number of flagella, these can be divided. Bacteria can be divided into 6 types. First one is atrichus. Example is monococcus. As seen in the previous class, they are spherical bacteria which do not have any flagella. Second one is monotrichus, which includes only one flagella at any end of the bacteria. Example is Pseudomonas and Vibrio. Then we have amphitrichus, which means the flagella is present in both the ends. Okay, one flagella is present in both the ends. Example is Nitrosomonas. Then we have cephalotrichus. Example is Corynebacterium, and here a tuft of bacteria is present in one end, or a tuft of bacteria is present in both the ends, and this type of cephalotrichus bacteria is also known as amphilophotrichus. Okay, which you will see that lophotrichus, which is the 6th type of bacteria, in that the flagella is present only in one side in a tuft. Okay, and the last type is peritrichus, where many flagella is seen all around the body of the bacteria, all around the cell of the bacteria. Example for peritrichus is E. coli. An example for lophotrichus is Salmonella. Examples are important and you need to keep them in mind. Next we come to nutritional diversity in bacteria. So as we have seen before, that bacteria indicates or bacteria showcases the highest dietary variation seen in kingdom Monera, or seen in among all the kingdoms, and bacteria may be autotrophic or heterotrophic in nature. Okay, so first group is autotrophic bacteria. These include bacteria which can prepare their own food, okay, with the help of photosynthesis or chemosynthesis. Okay, so when the bacteria uses light to fix carbon dioxide, or when the bacteria uses light as a source of energy for photosynthesis, then such bacteria are called as photoautotrophs, meaning they are autotrophs which use photons or light energy to perform photosynthesis. And when they use chemicals, inorganic substances or inorganic substances most probably, to fix carbon dioxide, then such bacteria are called as chemoautotrophs, autotrophs which use chemicals to form photosynthesis. Okay, so bacteria use a special type of chlorophyll in order to perform photosynthesis, because the pigment is a necessary component of photosynthesis. Okay, so here we have two types of bacteria: green bacteria, which are green in color. Obviously, they consist of a pigment called as bacterioviridin, okay, which is green in color, which gives the bacteria green color. And then we have purple bacteria, which has the pigment bacteriopurpurin, okay, purple purpurin, okay, which gives the bacteria purple color shade. Okay, so both of them are necessary for photosynthesis. Okay, and they are present in different bacteria. So based on this only we have green sulfur bacteria, green non-sulfur bacteria, purple sulfur bacteria, purple non-sulfur bacteria. Okay, so all these photosynthesis is anoxygenic, meaning it does not end up with the release of oxygen like in plants where carbon dioxide is taken and oxygen is released out. No? Here the carbon dioxide is taken but oxygen is not released. Other than oxygen, some other gas can be released based on the chemical used. Next is chemosynthetic autotrophic bacteria. Here they use chemicals, inorganic substances such as nitrates, nitrites, ammonia, and use the released energy from such chemicals to produce ATP or for ATP production. Okay, they help in the recycling of nutrients such as nitrogen, phosphorus, iron, and sulfur. So some of the groups of bacteria under this category include nitrogen bacteria. Example is Nitrosomonas, okay, which uses nitrogen as a source of energy. Then we have iron bacteria. Example is Ferrobacillus. Sulfur bacteria. Example is Thiobacillus. Hydrogen bacteria. Example is Hydrogenomonas. And methane bacteria. Example is Methanococcus. Okay, methane bacteria are not to be confused with methanogens from Archaebacteria. These are eubacteria which utilize methane for chemosynthetic or chemosynthesis. Okay. The next type is heterotrophic bacteria, which utilize food or energy from other organisms. Okay, so they are the most abundant in nature. Majority of them are important decomposers, and heterotrophic bacteria can be divided into three further groups. We have parasitic bacteria such as Vibrio cholerae and so on. We will see in the next slide all the examples of parasitic bacteria in plants and animals. Then we have saprotrophic bacteria. Example is E. coli, Pseudomonas, and so on, where they feed on dead and decaying matter, organic matter. And then we have symbiotic bacteria. Example is Rhizobium, which are in symbiosis with leguminous roots of leguminous plants. Okay, you can see the nodules. So those are bacteria in association with the plants. Okay, symbiotic association with plants. Okay, so next let us come to the classification of bacteria based on the nutrition. Okay, so based on the nutrition, bacteria can be classified into autotrophic bacteria and heterotrophic bacteria. Autotrophic bacteria and heterotrophic bacteria. Autotrophic bacteria can again be divided into photoautotrophs, meaning they take light energy for photosynthesis, and chemoautotrophs, which means that they take energy from chemicals in order to perform photosynthesis. So first let us see photoautotrophs. Again, as I mentioned earlier, photoautotrophs can be divided into sulfur bacteria and non-sulfur bacteria. Sulfur bacteria may be again divided into purple sulfur bacteria, which have the chlorophyll, the pigment bacteriopurpurin, and green sulfur bacteria which have bacterioviridin. Similarly, in non-sulfur bacteria we have purple non-sulfur bacteria and green non-sulfur bacteria. The examples are given. For purple sulfur bacteria it's chloromatium. For green sulfur bacteria it's chlorobium. For purple non-sulfur bacteria it's rhodospirillum. And for green non-sulfur bacteria it's a chloronema. Okay, you see that there are everything is very similar, so you need to be very careful in memorizing here. Okay, in Neet questions come with respect to chlorosium and chlorobium. Okay, so you need to be very clear with this. Then we have chemoautotrophs. Chemoautotrophic bacteria are again divided into 5 types. We have nitrifying bacteria, example Nitrosomonas; sulfur bacteria, example Thiobacillus; iron bacteria, Ferrobacillus; hydrogen bacteria, Hydrogenomonas; and methane bacteria, example is Methanomonas. Okay, all these use the particular chemicals in order to perform photosynthesis. And the second type of nutrition is heterotrophic bacteria, which includes three groups: saprophytic bacteria, symbiotic bacteria, and parasitic bacteria. The examples we saw as before. Next is examples of bacteria, parasitic bacteria in plants and animals. So I have mentioned the list of bacteria and the diseases which the bacteria causes. Okay, this comes with respect to Neet, and I have only mentioned those bacteria whose name do not, whose disease name does not, coincide with the name of the bacteria. Okay, for example, normally. Xanthomonas citri and citrus, okay, is similar. So you need to know the genus of the bacteria, which is Xanthomonas, and citri, which causes citrus canker. Okay, so this is an easy way. So once you know the species name, you can determine the name of the disease. But then there are other bacteria whose disease name are not similar to the species name, okay, or the bacteria name. We have Agrobacterium tumefaciens, which causes crown gall, okay, which is different. Then we have Vibrio cholera, this is animal disease. And then we have Mycobacterium leprae, causing leprosy. We have Helicobacter pylori, which causes peptic ulcer, okay, which then we have Yersinia pestis, a very important example, and it has come in Neet several times, which causes plague. Then we have Bordetella pertussis, which causes whooping cough, again another important example. And the final one is Treponema pallidum, which causes syphilis, which is a sexually transmitted disease, STD. Yes, then we have reproduction in bacteria, which is the final topic from today. And in bacteria, we see that there are two types of reproduction: asexual reproduction and parasexual reproduction. Okay, there is the absence of sexual reproduction in bacteria because there is no formation of gametes, or there is no fertilization of gametes. Okay, but a similar process is seen, and that is why sexual reproduction here is termed as parasexual reproduction or genetic recombination. So let us look into each one of them. Asexual reproduction, the first example is binary fission. Binary fission is nothing but a division of cell into two. Okay, this is the most common mode of reproduction. So here what happens is, let us look into the diagram. There are 4 steps in binary fission. The first step is that the prokaryotic parent cell initiates a replication, meaning the nucleoid present in the bacterial cell. One second. Yes, the nucleoid present in the bacterial cell attaches itself to the plasma membrane of the bacteria. Okay, as you can see, there is an attachment site here. Okay. Then what happens is replication starts and the DNA is replicated. Once the replication finishes, the third stage starts, which is the elongation of the cell and formation of the cross wall. Okay, so once the cross wall formation is completed, then the two daughter cells separate. So from one mother cell we got 2 daughter cells. So this is binary fission. Okay, important points include that the binary fission here, the cell division is amitotic, meaning there is no formation of spindle fibers as seen in the general cell division seen in eukaryotic cell, no? Like mitosis in human cell. Okay, here it is called as amitotic type because there is no formation of spindle formation. Okay, then we have that because of this, bacteria do not show any alternation of generation. Okay, there is no alternation of generation between haploid and diploid stage. It is always in the diploid stage only. Okay, and then we have, they are always in the haploid stage and they are never diploid. They are never in a diploid stage. Okay, all this because of the absence of meiosis and syngamy. Okay, Syngamy is fusion of gametes. The second type of asexual reproduction is endospore formation. Endospore formation is nothing but the entire cell of the bacteria condenses to form a thick walled spore. Okay, such a spore is called as endospores. Endospores are only formed during unfavorable conditions when there is less food or water, less food and water. Okay, so yes, as I said before, they are thick walled and they are formed during adverse conditions. Example is Clostridium and Bacilli. Okay, as we can see that there is a formation of a 4 spore in the mother cell, and then as soon as the spore is formed, the DNA in the mother cell disintegrates and thereby releasing the spore by breaking the cell. Yes, the second type of reproduction seen is parasexual reproduction. As I said before, since there is no fertilization and gamete formation seen in bacteria, true sexual reproduction is missing. Okay, although a primitive DNA transfer is seen as a kind of sexual reproduction. Now, this primitive transfer is of 3 types: conjugation, transformation, and transduction. So, let us look into the scientists who first discovered these processes, the organism in which they first discovered this, and the year. All this is important with respect to Neet. Okay, so for conjugation, it is Lederberg and Tatum. They first observed this in Escherichia coli or E. coli in 1946. Transformation was first proposed by Griffith, by the famous Griffith's experiment, was seen in Streptococcus pneumonia in rats. No, he did the experiment which was performed in 1928. And the final one is transduction, which was proposed by Zinder and Lederberg in Salmonella typhimurium. Okay, which was seen in 1952. Okay, now let us look into each of these processes in detail. Conjugation. Conjugation is nothing but a transfer of DNA from one bacterium to another bacterium with the help of cell contact. Okay, now how does this cell contact take place? The bacteria comes close to one another, and the pili which is there on the bacteria fuses with each other and forms a conjugation tube. Okay, what does it form? A conjugation tube. Okay, so after that, what happens is the bacteria which has the F plasmid. F plasmid is also called as the F factor or sex factor or fertility factor. Replicates the F factor in its cell, and then transfers the one copy in the other bacteria. So what happens? The DNA is transferred. The F plasmid or DNA is transferred from one bacteria to another bacteria. Okay, the bacteria with this F factor is called as F plus or the donor bacteria, and the one which without the F factor is called as F minus or the recipient cell. Okay, here since pili acts as a organ of attachment, it is termed as the sex organ of the bacteria with respect to conjugation. Yes. Next one is transformation. Transformation is one of the simpler forms of DNA transfer. Here the transfer of DNA takes place in a medium. Okay, a medium can be a liquid medium, generally wherever the bacteria is present. So what happens is you can see that a red color foreign DNA or from the medium, if the cell is ready, and we call this a competent cell. Okay, if the cell is competent enough to take the bacteria, it takes in the foreign DNA and it either incorporates it into the genomic DNA or lets it exist in the bacterial cell as a plasmid. Okay. Yes, the third type is transduction. The transfer of DNA here takes place with the help of a virus and especially a bacteriophage. Okay, normally bacteriophage. Bacteriophage that is released from the donor cell. Once the bacteriophage which is released from the donor cell helps in the transfer of genetic material. So how does this happen? I will explain. So let us see here that a bacteriophage has infected a bacteria. Okay, and ejected its DNA into the bacterial cell. Now it can go either two ways. So let us see the two life cycles of the bacteriophage. The first one is lytic cycle. In lytic cycle, what happens is the DNA of the bacteriophage controls the DNA of the bacteria and orders it to produce or synthesize phage particles or proteins which help in the formation of bacteriophage. Okay, the bacteriophage is made up of two parts, no? The capsid, or the what do you call it, the outer covering, and the DNA. So the DNA orders the bacteria to produce more of the phage capsid, the capsids. Okay, and once the bacteriophage is formed, all the materials are formed. The assembling of the bacteriophage takes place, and then they kill the bacteria and get out and go to infect other cells. Okay, and the other type is lysogenic life cycle, where the what do you call it, the DNA of the bacteriophage gets incorporated into the DNA of the bacteria and forms a recombinant DNA called as prophage. Very important, called as prophage. Okay, now as and when, and this becomes dormant now. So as and when the bacteria divides, the prophage also divides. Okay, and thus the increase in the number of bacteriophages taking place. And then when a external stress is given, or if a trigger is given, that time the bacteriophage, the DNA of the bacteriophage is removed or cut into several parts, and then again it again orders the, what do you call it, it goes into a lytic cycle, orders the bacterial DNA to produce phage particles, and then kills the bacteria. So here what happens is, during lysogenic cycle, when the DNA of the bacteriophage gets separated from the bacteria, there is a chance that the some of the bacterial DNA also comes along with the phage DNA. So because of this only, when the bacteriophage formation is happening in the lytic cycle, after that when the formation of the lytic cycle is happening, yes, when the formation of the bacteriophage in the lytic cycle is happening, that time some part of the bacterial DNA also is incorporated in the bacteriophage DNA, and this is how, when it goes and affects the other bacteria or infects the other bacteria, the DNA from this bacteria is transferred to the DNA of the other bacteria. So it's a very long process, and it's a very complicated process, and it is totally dependent on chance. Okay, so when this happens, there's a transfer of DNA from one bacteria to another bacteria with the help of virus such as bacteriophages. So with this we come to the end of today's class. My name is Hussain. Thank you and have